Thank you. Well, good afternoon, everybody. I hope you all have a wonderful day. I'm going to tell a story.
Yeah, we do need the mic.
I'm going to tell you all about the research participation opportunities there are in the Cavernous and Angioma space. But I kind of wanted to start with a funny little story that started my career in this space. 13 years ago, I was a newbie graduate student at Duke University. I was in my lab, and it was just a regular day, we're doing our thing, when a postdoc comes running down the hallway, like screaming. And this was a very calm woman. She was very mousy, quiet, kept to herself. She was excited beyond belief because she had found the CCM preview. She found it. Like, it was a huge day. My boss came in and said, if this isn't the most exciting day of your life, you shouldn't be in science. Well, I'm still in science. It was an amazing day, and it just changed things. You know, it was 13 years ago. Just imagine the space. Like, within 13 years, I know everybody wants to go faster, funny, but within 13 years, we identified a gene, we found out what it's doing, and now at the end of my talk today, it's going to be about clinical trials. So we are making tremendous progress. It could be faster, but it's hard. So just takes a lot of. So with that, these are our targets. This is accurate as of yesterday, well, 2 days ago, actually. These are all of the drugs or druggable targets that there are in the CCM pipeline right now. This is on our website, and I think you have a copy of it in your pamphlet. As of 2 days ago, there were 10. Now there are many more. The last 2 days of science upstairs has just been amazing. There's a lot more going on right now. All very, very new, very early in the preclinical space, meaning that it's in mice or in cells or something that needs to be worked out. So there's a lot more work to do, but what we're getting there, and this is really cool. So this slide summarizes the published data. Like I said, most of which is still preclinical work. So considerations for clinical trials for the CCM space. How do we move all those drugs to clinical trials? As Rustin so nicely told us this morning, it's important to take our time, do it well, collaborate with one another, and really have thoughtful, proper trials done. So how can we do that with CCM? What are the knowledge gaps? What are the things that we need to learn in order to get us to the next stage? One of the considerations is that we're a rare disease population, so it's always going to be challenging to find symptomatic people to fill trials. So this is where kind of Dr. Wadd's lunchtime speeches, we all need you to participate. We need the community to get behind us, and we all need to be in this together. And then our knowledge gaps are really, what are we going to measure? When we do clinical trials, what can we measure that will be effective as an outcome? Because everyone's experiences different symptoms. Some people have much, some people don't. Some people have headaches, there's a full spectrum of clinical variability that can muddy the waters a little bit. So what to measure? We talked about hemorrhage an awful lot. I'll talk about that later. I'm also going to talk about quality of life. And then some things that are most at the forefront right now are these imaging biomarkers, which kind of acts as a surrogate for an actual hemorrhage event. So this is also in your packet and on our website. This is kind of a schematic flowchart of all the different research opportunities there are for participation right now. Left-hand side of clinical drug trials, we see there's only one, the torbicide patch epic trial, which we'll talk about at the very end. I want to start on the other side of the research studies, because the research studies, while they're not clinical trials, they're still extraordinarily important because they're working towards filling in each of those gaps. So we'll start at the top. The first one is our DNA and tissue bank. So have you had a recent or upcoming surgical resection? If the answer is yes, please contact me, and we'd love to enroll you in our DNA and tissue bank. So DNA and tissue bank is a fantastic resource that Angioma Alliance offers to the patient, or to the research community, excuse me. So a lot of our researchers are not clinicians, are not surgeons that have access to tissue. The tissue samples are extraordinarily valuable. The information we can gain from that is incredible. I was a personal heavy user of biobank when I was in graduate school, and it got me through my thesis studies and to where I am today. So I'm pretty passionate about this. So what we do is we collect clinical records, we collect imaging records, tissue, if you've had surgery and it's left over after surgery, we collect that, and blood sample for DNA, and we can provide those materials to the researchers. They have to write a proposal, they have to be approved, and they never receive your identifiable information, like your name or address or anything like that, but they know all the clinical information about you. So we provide it to the researchers, and so how can we help? So if we are currently enrolling, so we're having a surgery, and who's not having a surgery coming up, please contact me. I'm the person to get in touch with. And really involvement is just talking to me on the phone, signing a bunch of paperwork, and allowing us access to those records. And then of course following through and getting your blood sample. We'll send you a kit for the doctors and your blood sample done. It's pretty much it. It's pretty straightforward and not too big and expensive. So we'll pause because I know I'm a fast talker and I get smart. The next research study, do you have a brainstem lesion or have had one removed surgically? So Dr. Fleming may have talked about this morning. At the Mayo Clinic, she's really interested in how to best counsel her patients for brain stimulations. There's not a ton of data out there, and it affects your quality of life. There's a lot, like I said, it's extremely variable clinical outcomes, but there's also quality of life that's affected too. Are you able to work? Are you able to drive a car? There's all these other factors that impact your life beyond what's in your medical records. So she's interested in learning about that. So it's a questionnaire, it's a survey. I don't have the email address up here, unfortunately, but it's on our website. There's an email address you can write to, and she'll send you the consent form, and if you consent to be part of the study, there's a number of questionnaires you have to fill out. I believe it takes about an hour to complete the questionnaires. But again, it helps with their body of knowledge about the current state of folks with brain stimulations and help us to counsel patients. I'm going to pass by. The next question is about hemorrhage. We're going to come back to hemorrhage at the end because there's a whole bunch of hemorrhage information. But I'll jump down to just cavernous angioma running in your family. So for folks that are familiar with cavernous angioma, meaning you either have a known germline mutation that you can hear from somebody in your family or acquired otherwise, or you have a family history with multiple lesions not associated with the TBI. If you fall into one of those categories, you would be eligible for the BVMC study. And the BVMC is the Brain Vascular Malformation Consortium study. And what this is, it's a huge multi-center study that covers 3 different vascular diseases. It covers CVM, obviously, HHT, which is hereditary hemorrhagic telangiectasia, and Sturge-Weber syndrome. All of them vascular underpinnings, and I think it's 15 different centers that are working together to study these. The CCM project is focused on natural history and endpoint definition. So we're collecting longitudinal data on people on a daily basis to see their clinical status, how things change, hemorrhage. So we really have a great understanding of what the disease normally looks like. Because we need to know that for clinical trials. We need to know what to expect, so you can know if we expect you to go from here to here within a year, and we treat you, and you don't make it to this point, that's good. But we need to know what this point is, and knowing that natural history is really, really important. And we have some of that data for this body. And so BVMC is a great project. Been going on for 9 years now. It comes in 5-year cycles. So we're in the second 5-year cycle. We just applied for a 3rd cycle. So we're working on that. So we're developing a clinical history, natural history, biomarkers is the other big thing that comes out of that. And really what they're looking at is, why is there so much variability? Even within families, we're trying to find the modifiers of disease. There's genetic modifiers, environmental modifiers, other things that might be affecting why you versus somebody else within your family is so differently affected. Enrollment right now is happening at the University of New Mexico, Baylor Neurological Institute, University of California, San Francisco, and Angioma Alliance will also recruit folks. So if you recruit through Angioma Alliance, it's all remote. Again, talk to me on the phone, we'll send you a blood kit, sign up a bunch of paperwork. Some key findings, so it's an ongoing study. There have been a lot of, one of the big things that have come out of it is genetic risk factors. So we know that inflammation is really important to CCM disease. So they've looked for genetic modifiers throughout the genome. So not genes, not like the CCM genes. They're looking for just small changes in the genome that might alter or modify the phenotype. But they found there were inflammatory and immune response to genes that were associated with differences in disease severity. And interestingly, the BVMC data supported the microbiome research in humans. So the microbiome project that came out last year was a really cool story out of mice, where folks at Penn were able to show that bacterial signals that come from your gut travel through the blood, send another signal, and it actually changes the way the lesions form in the brain. Which is really cool. Like that possibility of maybe altering the gut microbiome and affecting immune development is just mind-blowingly cool. So, but the nice thing is, those mice, and we always, are kind of worried mice are people, so we never want to jump to strong conclusions. I don't want you to change your microbiome, don't do anything crazy, just be healthy right now. But there was an inkling of really good data. So the signal that the bacteria sends is received by this TRL4 molecule. It doesn't really matter what it is. But the point is, BVMC data found changes in the TRL4 gene that correlated with disease severity in humans. So there is a reason for us to jump and take that next step and go to the human research. So that's where we're at right now. We've collected between 3 or 4 different sites, 4 different sites. We've collected 90 samples of human fecal samples. I've never spent so much time talking about food in my professional career. But we're doing it, and it's under analysis, and it will to be continued. So the work isn't published yet, but to be continued on that story. So clinical endpoints. When doing a clinical trial, you need to know something to measure. We'll talk about something. What do you measure? The FDA likes you to measure things that relate to clinical health. So hemorrhage, seizures, focal neurological deficits, this whole laundry list here. Hemorrhage is a biggie that we're going to talk about later. But Angioma Alliance is really interested, again, because of this variability, how can we kind of survey everyone? And quality of life is a biggie for us. I think that quality of life is really, really important. There's a bunch of functional outcome surveys out there that are already validated tools and used in trials. There's this thing called the NeuroQual, which is the Neurological Quality of Life Survey. So you take the survey and it covers anxiety, it covers how you feel about yourself, it covers all these different spectrums, but it's very broad, it's neurological. CCM is a neurological disease, but we have maybe a more reined in need for a survey such as that. And the modified Rankin scale is the MRS, and that's basically, it's a 0 to 5 scale of, how are you feeling, slight disability, a little, slightly more disability, so it's not terribly descriptive. So what we want to do is we want to follow along with this quality of life idea and really develop a formal CCM health index. So it's a survey-based tool that's specifically focused on CCM, and it can actually give us a score at the end, say, to rank everybody. Would you rank your health status at one point in time? So we're working with Recursion Pharmaceuticals and a group out of the University of Rochester, who's they're pros in this, they make these things all the time. And it's a very kind of complex qualitative research project that you have to undergo in order to develop this thing. It's not like we can't just sit down and draw up the survey ourselves. You really have to put a lot of thought and research behind this. But again, we're going to need participants at varying levels. So how do you develop such a thing? So you start by doing patient interviews. So the qualitative researchers at Rochester will reach out to probably 10 or 20 people and just sit down and have focus groups. Like, what are the issues that are important to you? Everybody will have a different answer, but they're going to kind of tease it out and see what are the most relevant issues to our patient community. And then they go to a second survey of patients and kind of reiterate this a couple of times and develop a tool. They develop a long questionnaire, and then it goes through various iterations of testing and validating, and there'd be a lot of patient input, and we really need folks to take this questionnaire until it's validated at the end. And there's a true validation process that they have to go through, can be FDA qualified to be a certified clinical outcome measure for clinical trials down the road. So I'm really excited about this. It's a 2-year project, and we're about to get started with that. So look out for this as another research opportunity in the very near future. So the hemorrhage. Hemorrhage is certainly clinically relevant, right? Nobody wants to have a hemorrhage. However, not everyone experiences hemorrhage, so it becomes a challenging thing in terms of recruitment to have a study solely based on hemorrhage. But we're working on it. So under the research studies, have you experienced hemorrhage kind of in the middle there within the last 12 months? If the answer is yes, there's this new project called the CASH Trial Readiness Project. So CASH stands for Cavernous Angioma of Symptomatic Hemorrhage. And why symptomatic hemorrhage? Because folks who have had a hemorrhage within the last year are more likely to have a second hemorrhage. So if we just have a trial and say, hey, we need all comers, everyone who enroll in the trial, we're going to measure hemorrhage. Be such a rare event, you need huge numbers in order for the trial to make it statistically valid. So by focusing on CASH, Cavernous Angioma of Symptomatic Hemorrhage, folks within the last year, then it kind of narrows that window down. So all these sites are getting together. Mayo, the Mayo Clinic, University of Chicago, got it right now because I can't remember all these. University of Chicago, Johns Hopkins, University of Mexico, UCSF, Utah, and Barrow Neurological Institute, that is it. What they want to do is they're really working together to prepare for large multi-center trials. Like we can do small trials on our own right now, there's 50, 80 patients, but once we get to phase 2, phase 3, we want to see really large, probably international multi-center trials. There's 2 things that are really super important to prepare for something such as that. One is to analyze the patient population. Do we have folks out there who would qualify for studies and who want to be involved in studies? So they're across those sites, they're going to figure out how many patients they have who would qualify for such a study, and also validate these imaging biomarkers. So we'll come back to that. So trial readiness participation at one of those sites, what participation looks like is going to the site at baseline, you get an MRI, you get a clinical visit, labs are done, and you're followed up for 2 years. So it'd be 3 site visits at baseline, 1 year, and 2 year. And the research goals again are to identify and confirm patient cohort and then the imaging biomarkers. So what do the imaging biomarkers look like? This is a biomarker dynamic contrast enhanced quantitative perfusion assessed permeability. So we just call it permeability, so that's way too much. So what it is so on the right, you see that T2 MRI that you've all seen with the lesion in the yellow box. You can actually measure the permeability within the brain. So what they do is they give you an injection of gadolinium, which is a contrast agent. It goes in through an artery and then it perfuses into the brain. It takes time, and they can actually measure the rate with which it perfuses into the brain and within a lesion. And they can see the lesion activity essentially of how much permeability is happening in that lesion. And you can see the green spot on the left. It's quite highly permeable in that lesion. And lesions that are more active on the and then the other biomarker measures iron. So QSM, quantitative susceptibility maps, it measures the iron content, which is a surrogate for blood. So again, this is from the Chicago team. It's an imaging technique to measure iron. So both of these techniques have been developed by the University of Chicago, and they're used and validated at the University of Chicago. They're completely fine use for clinical trials there. However, moving them to multi-site, part of this, the reason that they're doing this project is to show that they are replicatable, so that the data can be compared and you're actually measuring the same thing at different sites. So clinical trials. There's different phases of clinical trials: phase 1, 2, 3, and then post-marketing trials. The phase 1 trial is a safety trial. So you probably heard from Tim at Recursion about safety trials. Small numbers, usually quick. You just want to make sure that your drug is safe to take in humans, not CCM patients. Phase 2 is once you start looking at efficacy. It continues safety monitoring, but you want to know, does this drug do what we expect it to do in a larger number of patients? For our disease, probably going to be 1 to 2 year with about 100, maybe 200 people, given it depends completely on what your endpoints are, but just kind of for relative ranges. And then phase 3 are your really large multi-center trials that are for long-term efficacy. So the trial that we're all talking about is a atorvastatin cash trial here in Chicago. So it's a phase 1/2. So what does that mean? So it's atorvastatin. Atorvastatin is a statin drug. Folks take it all the time for cholesterol reasons. We know that the drug itself is safe. Generally, with a new chemical entity, not generally, always with a new chemical entity, you have to do a purely phase 1 trial to show that it is safe to take in humans. With this, only phase 1/2, because we know it's a safe drug to take. However, we know it's safe in the CCM population. Likely yes, but we still need to test that safety aspect. And then the phase 2 part is see, is it effective? Does it actually block the hemorrhage like we're predicting it to? And the outcome measurement is going to be that QSM iron sensitive imaging sequence at the University of Chicago. So it's a single center study at the University of Chicago because that's where we know that biomarker works. So I'm sorry, I'm a scientist. I had to throw in one sciencey slide. My apologies. You don't have to care about the details, but I just wanted to go through why we think atorvastatin is going to work and what's interesting here. So what you're looking at are 2 blood vessels. So this is the junction between the blood vessels. And you see there's all these different molecules that really link those junctions together. And then over here we have the CCM complex, which regulates these because of this, Rho kinase. So Rho kinase, or ROC as we call it, that's what we're targeting. By targeting Rho kinase, we can shore up these junctions, because once you lose functionality of one of the CCM genes, we lose the junctions, and that's where the bleeding comes in. So what they've shown is that in mice, atorvastatin, so this is a mouse with CCM3 mutation, and it's got tons of lesions. I mean, these mice are they're a hot mess. We have tons of CCM lesions, placebo treated. And then down here, this is a histology. So this is a close-up view of one of the lesions, and the blue spots are all the iron. So this is a bleeding lesion. But when treated with atorvastatin, many fewer lesions, smaller in size, and no more bleeding. So this is really awesome evidence that's leading us down this road to clinical trials. So they're expecting the statin therapy to restore junction to people, and then hopefully see a decrease in QSM signal for iron in the brain. So it's now enrolling. We've got 4 folks enrolled already, but they need 80. So it's going to be the 80 folks who've not had surgery, who are symptomatic within the last 12 years, for reasons we just talked about, and also have not been on 12 months, last 12 months. And no statin therapy within the last 12 months at all. And travel stipends are available for folks traveling from out of state. And as Rustin told us this morning, CCMs with part of bleed are like 10 times more likely to bleed again. So again, just reiterating this whole catch and why we're focusing on folks with hemorrhage in the last 12 months. And what does it look like? Study participation, it involves 3 visits to the University of Chicago, very similar to the trial readiness protocol, the only difference being that you can drug or placebo, so it's placebo-controlled, randomized placebo-controlled. 3 visits to Chicago at baseline, year 1, year 2, when you go and do a neurological exam, physical exam, and have labs done, and then you have all the research MRIs done, and follow-up phone calls basically every 3 months in between, to see how you're doing, if there's been any adverse events, or if you need to change your dosage, or anything like that. And at the University of Chicago, contact Christina at AT Cash in Chicago. Again, it's on our website if you can't get it here. And join our patient registry. That's the other big thing, is our patient registry is an awesome tool that we use for recruitment. We've had it up since 2009, and right now it's undergoing kind of a massive overhaul to upgrade it, make it more friendly, make it friendly for researchers to use, and it's really just going to be a great communication tool we collect enough information about you to know which studies you may or may not be interested in, eligible to participate in. So I have a study that I need 40 women from the state of New York, and between this ages, I can search for those and let those people know what trials are out there. And you can also, on the back end, you can look at all the aggregate data and see how many folks enrolled have multiple lesions, how many folks have C1 gene mutations, or all these different things. It's always identified from your end. From my end, I can see who folks are, but from your end, you can see the identified data. And that will be rolling out soon. It is currently out in its old version, but the new version in the next couple of weeks. And I'm sorry, I have a fast talk. I got through this really quickly, but this is my final slide, just to drive home that without you, there can be no cure. And I really love this quote from the Center for Information and Study on Clinical Research Participation. It says, behind most every treatment, medicine, therapy, and medical device are millions of people who are volunteers to take part in clinical trials. So it's just super important to get out there and volunteer. I'll take questions.
I think there was a question earlier about imaging biomarkers
Yes.
in clinical care. Yeah, I was just curious. It seems like my question might have been answered, but I was curious about imaging biomarkers, if that's available to the general population, but it sounds like it's just at the University of Chicago.
Right now, they're just at the University of Chicago. New Mexico is also doing a different permeability imaging biomarker. But for research, so you have to sign a research consent form in order to do it. So it has to be one of the research sites that are actually involved. So if you go to one of those trial readiness sites, they have them available. They're working on them, so you can definitely do it there.
When you get an MRI, they typically ask you, who do you want to send it to, et cetera. Would you recommend that we also add you or your office, whatever, as one of the recipients, or is that just extra data that you don't really need and you'd rather have it be done elsewhere?
It can be uploaded to our new registry. You can upload things there. You can also, if you join the BDMC or you join our biobank studies, then we can take it. I wouldn't want it just directly sent to you. I want to definitely talk to you first. BDMC is not open to international participants right now for regulatory reasons. But Health Index, I suspect that'll be a yes. I've not seen the consent form for that yet, but I believe that it should not be a problem as long as the relationship percentage is in English or if you're.
I'm new to my brain. I probably very got it. Since I have to still love it as part of it. But you mentioned a couple months ago, well, you've had a brain bleed or in the past 12 months.
Yep.
So I had the MRI scan back over the summer, and I still have not yet had a doctor actually explain to me what is going on, which is haunting to me. Haunting. So I did finally tease out from a few people that it did bleed. It had bled multiple times, and that you can tell that it's bled multiple times because of the aging of the blood. So how there's a way that, and the doctor that didn't share that with me, there's no way to tell when it was. But clearly there is.
Right.
You want people who have bled. And
So there is a definition for symptomatic hemorrhage. And what it means is you have a symptom, you have a seizure, or you have a massive pain, or you have a symptom, you go to the hospital and you have an MRI done, and the MRI shows imaging along with your symptom. So that's what a symptomatic hemorrhage is. So
the MRI for me would show new blood, and that's how you would tell. But I can't look at the
Unless you happen to have your MRI and something else happened to you, someone would move that happened and it's correlated in the record.
Yeah, I mean, something wrong. Yeah, okay.
I mean, so when we're talking about the symptomatic hemorrhage and making that clear correlation, what about like for those patients like my daughter who have, it's become like a global assault, because there's so many innumerable lesions, and if you scan her on any given day, there's bleeding within the lesion. Sometimes there's bleeding without the lesion. So does that mean that someone in her situation really would never qualify? And she's had, as you know, multiple surgeries.
Sure, no, absolutely.
Yeah.
That would be at the discretion of the PIs of the trial. That's a doctor-wide question for sure. Because the definition of hemorrhage is really, Rustin wrote it up several years ago, and it's really a firm, hard definition that they're trying to follow.
Right.
I suspect that they could probably find one for her that made sense along with her medical records. But it would be a PI question, or it would be the lead study investigator who could answer that. And then he would make the decision whether or not she could be enrolled or would be eligible for the study.
Okay. And the other question I had in terms of the biomarkers, is that hardware specific or software specific, like what they're doing at University of Chicago? What is different about their MRIs? I mean, we'll be there next week.
Yeah.
So
I don't frankly
Again.
understand all of it, but
Okay.
it's all post-imaging. There's a lot of math that goes on, and like they're calculating things afterwards.
Okay.
And then I believe there's also proper settings that need to be done. I mean, it's doable across sites, but they just need to make sure everything is done exactly the same way.
Okay. Yes.
So of those sites that you had put up that they're going to be conducting trials eventually.
Yep.
If we've been to one of those, will they just contact us if they were, do we have to go and contact them?
It would not hurt to be proactive and contact them yourself. They may at some point, like if they're lagging behind in enrollment, they may reach out to their list, but I would contact them. Thank you, guys.
Thank you.
We have a celebrity here, Maggie. And could you come up and explain what folks are going to see and what you've been up to? Oh, okay, sure. This is Maggie Whittam. Those of you who are on Facebook are very familiar with her, I'm sure. And she is going to be showing us a video of a project that she has in the works and just wants to share it with us. Just a second. Maggie, did you want to tell us?
Yeah,
So,
hi everybody, my name is Maggie Whittam. I had a brainstem cavernous that I did not know was there, and it hemorrhaged Christmas 4 years ago, very, very badly, and I had to have brain surgery, which I got at Johns Hopkins, and they did a great job, and my surgeon was Dr. Judy Fong. She's amazing, and I'm doing okay, right? I'm here, right? And I was an actor before I got sick, and I really want to do something meaningful and powerful with my story, and I want to make a film about surviving my stroke and my life afterwards, and about disability and what it means. So this is just a little 2-minute teaser about this documentary that I'm going to make, and I hope you enjoy it. So we want to make a 90-minute film that's that good. So we'd love your support if you're interested in helping us out. We're crowdfunding right now. Just go to the website. Thank you.
How many of you purchased art from the art auction? We want to get a quick photo of Yana and Maggie, who are artists that were participants in the art auction, and the folks, and it doesn't need to be right now, but and the folks who actually purchased art, so that the next art auction we have that photo to be able to use to promote it. So maybe before we go into our small groups, we can just have those folks go over there. Does that sound right? Okay, go over there and we'll take a quick shot. Okay, so small groups. We have, I believe we said we have 5 of them. Maybe 6 of them. We have epilepsy, headache, other. If you don't feel like you fit in a category, that would be the good one to go to. And that's with Susan over here. Candy has the brainstem group, and we'll move things around a little bit so that we have a little bit more space. Tracy over there is the spokesmodel for the parents group. Jackie in the back over there has sinus with multiple lesions. If you have a hereditary form and you have multiple lesions. And then Robin has the caregiver group. They're going to spread out a little bit so that everybody can have a space. I think working around the table is not a bad thing. But certainly we're going to move some chairs to the other sides of tables.